Hi, my name is Kolberg, and I studied if Hippodamia convergens were more effective at removing Acyrthosiphon pisum than a chemical containing imidacloprid. Hippodamia convergens is the scientific name for ladybugs. Ladybugs are small beetles that come in many different sizes and colors. Their eggs grow to half an inch long in just 2 to 3 weeks, then pupate into adult ladybugs. Before becoming an adult, the larvae will shed its skin multiple times and eat around 400 aphids. An adult ladybug might eat 5,000 aphids during its lifetime and live 1 to 2 years naturally. However, there is a chance that it won't live its full life because it could get eaten by a predator. Also, artificial chemicals for removing pests are harmful to ladybugs and couldn't show in their lifespan. Acyrthosiphon pisum are also known as aphids. They are very small sap-sucking insects and are one of the most destructive pests for temperate gardens. They are hard to remove and can kill plants. It is possible to repel these pests with essential oils, but ladybugs are a more practical solution. The eradication of all aphids is harmful to the ecosystem. The chemical imidacloprid is an insecticide that acts upon the central nervous system of insects and was made to mimic nicotine. It works by disrupting an insect's nervous system by hampering the ability to send signals. Plants absorb imidacloprid to the roots in the soil and then pass it on to the insects or plants. This is an efficient way of removing aphids, but is it really the best? Farmers, planters, and everyday gardeners have a common problem that is stressful and difficult to deal with. This problem is aphids, which are small pests that kill plants by sucking the phloem from inside. Aphids multiply rapidly and cause infestations, which makes them a hassle to remove. Because of this problem, I was interested in finding out if Hippodamia convergens were more effective at removing Acyrthosiphon pisum than imidacloprid in the chemical powder. The ladybugs will do a better job at removing the aphids than the imidacloprid in the chemical powder. This is because the Hippodamia convergens directly eat the Acyrthosiphon pisum, while the imidacloprid takes time to go into effect. All groups had a plastic tank, 100 pea aphids, a pea plant, a succulent plant, 1½ inches of dirt. Specifically in the ladybug group, I had 75 ladybugs, and specifically to the chemical group, I had the chemical powder which contains the imidacloprid. The first thing I did was place the tanks on a flat surface near a small window and under overhead lights. Next, I prepared each tank with 1½ inches of dirt along with the succulent plant and watered it with a spray bottle. Then, to use the chemical powder correctly, I sprinkled it throughout the soil and in the pea plant of the chemical group. Since I could not separate the pea aphids from the pea plant, and I had to work quickly, I placed an entire styrofoam cup containing the pea plant and the aphids in each tank. To ensure that the aphids did not escape, I covered the top of each tank with a washed porous shirt to provide sufficient air. Next, I put the ladybugs into the ladybug tank and then noted my observations. Every day, I squirted water through the air vent and through the shirt to keep the environment moist. Since the plants and insects were not exposed to sunlight for 24 hours a day, I kept the lights on for half a day to simulate day and night. This light also kept the ladybugs warmer, since it was winter. During the first 5 minutes of my experiment, I observed that the ladybugs were devouring the pea aphids at a very fast rate, while I didn't notice any changes in the tank with the chemicals. 20 minutes later, most of the pea aphids were gone in the ladybug group, but nothing had changed in the chemical group. During this time, the pea aphids in the control group were spread all over the pea plant and were eating it. In all 3 tanks, none of the pea aphids were gone and were on the succulent plant. At the 30-minute mark, all observable pea aphids were gone in the ladybug group, and none of the pea aphids were killed in the chemical group. 3 days later, all observable pea aphids in the tank with the chemicals had died due to the imidacloprid. After a week, the pea plant in the control group was completely dead. In contrast, the pea plant in the imidacloprid tank more than doubled in size. The one in the ladybug group grew moderately. It took the ladybugs about a half hour, and it took the chemicals about 3 days to remove all the aphids. In the control group, the pea plant died a lot faster than the pea plant in the other 2 groups. Ultimately, my hypothesis was proven correct in that the ladybugs were more efficient in removing the pea aphids than the chemicals were. My tanks could have been bigger to simulate a more realistic environment. I had to keep the tanks closed and inside, which did not give them much natural sunlight. I could have used bigger sample sizes in my experiment, and I received more ladybugs than the 15 to 20 I had expected. In the future, I could include more plants and use a different species of ladybugs. Maybe I could use better equipment to get even more accurate results. Thank you. Have a nice day.